Hi, I'm Thea. I'm a PhD student from the University of Sheffield, and I'm going to present one of the chapters of my PhD today. So, males and females of the same species can be strikingly different, and this is not only in basic reproductive traits, but also in behaviour and morphology, as we can see from these drawings. And this is sexual dimorphism. So clearly the sexes often have very different evolutionary interests, and so are subject to very different selection pressures. However, males and females share the majority of their genomes. So the broad question we wanted to answer in this study was, how do you go from this shared genome to completely different phenotypes? So to date, there's been considerable focus on testing how genes with sex bias expression level encode sexual dimorphism. But the role of sex-biased alternative splicing in the evolution of sex differences has been overlooked and lacks empirical evidence. So in eukaryotes, introns are removed to produce mature mRNAs, and alternative splicing is a mechanism that enables different transcripts to be generated from the same gene. Sex-specific alternative splicing can ensure that certain exons, such as those with sex-specific beneficial functions, are retained in the gene product, and certain other exons, such as those that have sex-specific detrimental effects, are skipped and not included in the gene product. And therefore it might be an important mechanism for the evolution of sex differences. So these are the two most common types of alternative splicing events in vertebrates, which we've focused on in our study. So firstly, there are skipped exon events. So for example, in one sex, the isoform in the final transcript might include this blue exon, but in the other sex, it might be missing like this. And then there are MXEs. So for example, in one sex, the isoform in the final transcript might include this orange exon, but in the other sex, it might include this blue exon instead, but never both. So sex differences in splicing therefore have a huge potential to play a really important role in the evolution of sex differences. So we identified alternatively spliced genes across the autosomes using RNA-seq data from the reproductive tissue of the turkey, the guinea fowl, and the duck. So here we have a heat map and dendrogram for the gonad tissue for the orthologs of all three bird species. So each row is a gene, each column is an individual, and males are in blue and females are in red, and the green and purple colours represent the PSI value, which is a measure of alternative splicing. So patterns of alternative splicing cluster first by species in the gonad, which indicates that patterns of splicing evolve rapidly. And then gonad samples cluster by sex in all species, indicating that splicing varies substantially between the sexes. And we wanted to understand what drives this variation and the consequences for sexual dimorphism. So the three questions we asked in this study were, is alternative splicing associated with sexual dimorphism? What drives the evolution of sex-biased alternative splicing? And are sex differences in gene expression and splicing independent routes to phenotypic sexual dimorphism? So now on to the first set of results. So we asked, is splicing associated with sexual dimorphism? And to answer this, we looked at gradients of sexual dimorphism across turkeys. So turkeys exhibit extreme sexual dimorphism, and here you can see some traits that differ between the sexes. However, in the turkey, there are actually two male morphs, so there's the dominant and subordinate. And dominance among sibling males is determined by male-male competition prior to them becoming sexually mature. Males that develop the subordinate male phenotype still exhibit sexually selected traits such as iridescent plumage and long beards similar to dominant males, but they have less vivid head and neck coloration and less developed snoods and wattles, which is the bits on the turkey's nose and necks. But these features are still more vivid in colour and bigger than in the females. So subordinate males are male in phenotype, but they lie in an intermediate position on the sexual dimorphism continuum. So we compared patterns of splicing along this gradient of sexual dimorphism. So first, we identified genes with significant differences in splicing between the dominant males and females. And we then compared patterns of splicing, so the average PSI in subordinate males. And you can see from this graph that subordinate males exhibit intermediate patterns of splicing between dominant males and females. These numbers are the p-values. So then for genes with significant differential splicing between the sexes, we classified the alternatively spliced isoforms as either male or female biased, depending on whether they were expressed more highly in dominant males or females. So we can see from the second graph that subordinate males express male-biased isoforms in the gonad at significantly lower levels than dominant males, indicating that male-biased isoforms are demasculinized in subordinate males. And from the third graph, we can see that subordinate males also express female-biased isoforms at significantly higher levels than dominant males, consistent with feminization of female-biased isoforms in subordinate males. And these patterns are consistent with the phenotypic sex differences observed across morphs, where subordinate males occupy an intermediate position on the continuum of sexual dimorphism. Answering our first question. So our second question was, what drives the evolution of sex-biased alternative splicing? And there's lots of debate as to whether splicing is functionally important and whether most isoforms evolve under drift. And so our questions tie into this broader debate. So if you remember earlier, the heat map indicated that patterns of alternative splicing evolve rapidly. And we wanted to find out why this was. Specifically, we wanted to test whether this rapid rate of evolution was a product of sex-specific selection or genetic drift. In order to test this prediction, we used regulatory variation as a proxy for selection. So the more diversity of alternatively spliced isoforms, the more selection. When expression level differs between the sexes, selection should be strongest in the sex with a higher expression, as selection on loci increases with expression level. So we expect selection for sex-specific isoforms to be greatest in the sex with a higher expression level. Therefore, if the rapid rate of evolution of alternatively spliced genes is due to sex-specific selection, we predict a greater diversity of isoforms expressed in males than females for genes which are more highly expressed in males, so they're male-biased. And then novel male-specific isoforms will be free to evolve male-specific functions, while isoforms expressed in both sexes are retained to perform their original function. And we predict the opposite scenario for female-biased genes, which under sex-specific selection should exhibit a greater diversity of isoforms expressed in females. However, if the rapid rate of evolution of alternatively spliced genes is due to drift, we predict the greatest diversity of isoforms in the sex with the lowest expression, where selection is weakest, reflecting transcriptomic noise. And this is because there would be less purifying selection on lowly expressed genes. So to test this, we developed an isoform specificity index, which we called tau-AS, to quantify variation in isoform abundance per gene. So here on the y-axis of these box plots is the isoform specificity index, tau-AS, and low values of this index indicate an even representation of isoform abundance, and high values show that a single isoform is always expressed. We found that in all species, males show a significantly greater isoform diversity for male-biased genes, and females show more isoform diversity for female-biased genes. And there were no significant differences in isoform diversity between males and females for unbiased genes, which are in grey. And this is in line with our predictions under sex-specific selection. So this allows us to answer our second question. Our results are consistent with sex-specific selection acting on isoforms that are differentially expressed between males and females, driving the evolution of alternative splicing. So now we'll go on to our last question, which is, are sex differences in gene expression and splicing independent routes to phenotypic sexual dimorphism? And we looked at this because it tells us about the transcriptional architecture of sexual dimorphism. So for example, is each mechanism equally as important? Do they work together, or do they play distinct roles? Firstly, if splicing is a distinct route to the evolution of sex differences, we expect sex-biased alternative splicing to be more common in genes with similar expression patterns between males and females. And we looked at the overlap between sex-biased gene expression and sex-biased alternative splicing to test this. So genes which are sex biased in an expression level are in blue, and genes that are sex biased in alternative splicing are in orange. And we found that there is no significant overlap between differentially alternatively spliced and differentially expressed genes in the gonad in any of these three species. So they're largely different sets of genes, which suggested to us that sex-biased alternative splicing could be an alternate or a complementary route to sex-specific adaptation. So pleiotropy is where a gene performs several functions and affects multiple traits, and it's thought to hinder the evolution of differential gene expression level and limit the response to sex-specific selection. So genes with broad expression patterns, which is a proxy for pleiotropy, are known to be less likely to be differentially expressed. And alternative splicing might avoid pleiotropy and other constraints acting on expression level through the generation of distinct male and female isoforms. And we set out to test this. So we calculated tissue specificity, tau here on the y-axis, for unbiased genes in grey, differentially expressed genes in blue, and differentially spliced genes in orange. And low values indicate broad expression patterns, and high values indicate more specific expression patterns. So differentially spliced genes were found to have significantly broader expression patterns than differentially expressed and unbiased genes, indicating that differential splicing may be a route to overcome pleiotropic constraints, acting on genes that limit the evolution of sex differences in expression level. In addition to this, we looked at the rates of coding sequence evolution of differentially spliced and differentially expressed genes. So this is DNDS. Previous studies have shown that differentially expressed genes in these species have elevated rates of coding sequence evolution, and this is due to genetic drift. In contrast, however, we found that genes with differential splicing don't exhibit significantly elevated rates of sequence evolution in comparison to unbiased genes or genes that are differentially expressed between the sexes. And this is consistent with stronger purifying selection acting on the coding sequences. So this increased strength of purifying selection we find suggests that genes that evolve sex differences in splicing are under strong constraint at the coding sequence level. So taken together, these results suggest that when genes are subject to functional constraints, the evolution of sex-specific isoforms may offer a more viable mechanism than changes in expression level to achieve sex-specific functions. So to answer our last question, differential alternative splicing may present an alternate route to the decoupling of male and female phenotypes when sex differences in gene expression level are under functional or pleiotropic constraint. So I just wanted to very briefly touch on how I plan to take this work on transcriptional regulation further. For the last chapter of my PhD, I'm currently looking at alternative splicing and gene expression level in the same 3 bird species plus 4 more, which you can see in this phylogeny here. These species vary in their levels of sex-specific selection, which you can see here. And my aim is to see if each transcriptional mechanism differs in their potential to deconstruct genetic correlations between the sexes and tissues across broad timescales. I hope our results reveal the constraints acting on gene regulation over broad time periods. So with that, I'd like to thank my lab group, which includes my supervisor, Alison Wright, as well as Daniela Palmer and Peter Price, and my sources of funding, and Clara Lacey, who did the really beautiful illustrations at the beginning of the talk. Thank you for listening. I'll be happy to take questions later.